Hi, thank you for tuning in to learn more about my poster and listen to me go through it. This is Maggie Doolin from the University of Utah School of Biological Sciences, and I'm presenting my poster, Parasitism in the Gut Microbiome of a Mammalian Herbivore. And today I'm going to tell you a story about parasites in the microbiome. And we know that the gut microbiome performs essential digestive functions, especially across vertebrates. And specifically in herbivores, a couple of those functions include fiber degradation and plant toxin degradation, both of which are essential processes performed exclusively by the microbiome and not performed at all by the mammalian digestive tract. So we know that within mammals, the gut microbiome is essential for herbivorous digestion. We set out in this project to understand whether a disturbance by parasites within the system of white-throated wood rats in Castle Valley, Utah, actually inhibits this function in some way. Specifically, we wondered if wood rats with parasite infections would have reduced gut microbial diversity and function compared to parasite-free wood rats. And we hypothesized that this would be true. In figure 1 here, just to show you our sampling site in Castle Valley, Utah, the Opuntia cactus, this dietary, kind of big dietary component for these animals, that is also rich in dietary fiber and the plant toxin oxalate. Here's an egg from the feces of these animals showing the parasite, the hindgut nematode that lives in these wood rats commonly. And then finally, a picture of a wood rat itself. And in order to investigate whether parasitized individuals had a different microbiome diversity and function compared to parasite-free individuals, we live trapped wood rats in May and October 2019 and held them for a 4-day diet trial feeding only on Opuntia cactus, and then we released those animals. And so we took a white-throated wood rat here and Opuntia cactus that was powdered and dried and put them both into a metabolic cage for 4 days, continuously feeding these animals and tracking how much they were eating and drinking. And then at the end of the trial for 4 days, we collected urine and feces. Each day we collected urine and feces. And then we used these samples to diagnose parasitism and also to characterize the microbiome based on 16S barcoding, which is a common way to characterize microbial communities. And finally, we use them to assess microbiome function. And the nice part about herbivorous digestion is it gives us these nice functions of fiber degradation and oxalate, this plant toxin degradation, that are done only by microbes and not by the host. So it gives us a nice functional aspect of assessing the microbiome health directly. So when we got our results back, and just to go up to the results panel here, we found that gut microbial richness and alpha diversity do not differ based on parasitism or sampling date. And showing this in these two plots side by side here, on the left, we have the observed amplicon sequence variant richness, just the number of different unique 16S sequences that were seen in each individual microbiome. And sampling date is what this plot is divided up by, and then parasitism is coded by color. And we saw no significant difference based on sampling date or parasitism in the richness. And then here with Shannon diversity, taking evenness of the distribution of the microbial taxa into account as well, we saw no difference. So basically, alpha diversity and species richness, we're not seeing a difference based on either of these metrics. Then we move down to the middle plot here, looking at microbiome composition or beta diversity. And this is a principal coordinates analysis of Bray-Curtis measurement technique. And essentially what we're seeing here is that parasitism is coded by color and sampling date is coded by shape. And each different symbol represents one animal's microbial community, with closer symbols to one another being more similar microbial communities. And when we do the analyses on this ordination, we find that both sampling date and parasitism are influential in understanding the composition or the distribution of these microbiomes in comparison to one another on this plot. However, sampling date has a much stronger effect on this than parasitism does. So we're seeing an effect of both here based on beta diversity analysis. And finally, just looking at differential abundance. So if we're looking at differences in composition in the middle plot, at the bottom here, we're looking at specifically which ASVs, which of these 16S sequences are seen more in individuals with and without parasites. Just a summary table here showing that there are several more ASVs enriched in animals without parasites than those enriched in animals with parasites. But all of the families that have ASVs enriched in either group have important functions for the gut microbiome in wood rats, but also across animals. Some of these are also common to human microbiomes. And so here I have summarized the known family functions, just knowing that gut microbiota with critical functions are reduced in animals with parasites, since we're seeing this higher enrichment of several of these taxa in animals without parasites. So if you'd like to learn more about this work, you can scan this QR code with your phone. That'll take you to my website to learn more with some expanded results that we found from this project. And continuing in future, I'm just going to be doing those direct functional measurements of oxalate degradation, especially to look for differences in microbiome function, and also to investigate whether if we spike the diet with oxalate and don't give animals only plain cactus, but actually extra oxalate, will we see a difference in kind of an accentuated or exaggerated difference in animals with and without parasites compared to what we saw just on a cactus diet. So thank you for listening.